Hey, Meytal, how are you doing?
Hey, how are you?
Shalom. How is it going?
Okay, you busy Saturday morning?
That's good, right? Good to keep us also busy in this pandemic situation. So that's that's good. So how are things going for you?
Okay, Corona is annoying, like I guess everyone,
Yeah.
but it's as I wrote you, like I'm trying to open the second lab in Hamburg. So and I wanted to
Where in Hamburg? Is the University of Hamburg or what is it? You are not audible. Can you hear me now? Hello?
I don't know why
Yeah,
it's
now I
this
can hear you. Yes. So where in Hamburg? Which institute?
in
No, I can't. I don't hear you. Keep talking. Keep talking. Nope. Nope. Maybe is it because of my internet issues or
I think it doesn't want to hear my headphones. I don't know
now I
why.
can hear you. Now I can hear you. So maybe it's possible that my internet is a little bit slow. Are you able to hear me continuously?
Yes, yeah.
That means my internet is okay. So you are okay now. I'm hearing you. So where is in Hamburg? Which University of Hamburg or?
In Desi, the German synchrotron, where I tried
Mm-hmm.
headphones again. They don't want to disconnect, and I think
Yeah, now you are disconnected. Try without the headphone. Maybe direct use a computer microphone. Speak now. It should be okay.
yeah, if like this, you can hear me fine.
Yeah, now it's okay. Yeah, it's perfect. So you don't
Okay,
need the headphones.
okay.
So when are you joining in Germany?
So I'm in a process. I have a signed contract with EMBL
Mm-hmm.
for 5 years as a visitor group leader. But it doesn't give me the funding, so I can apply for external funding via EMBL and then manage everything. But it will take time, and like EMBL is not Germany, so they are not open to all the German
Correct.
funding available. So
Correct.
I'm trying to get like other sources of funding so I can, get some seed money to start the lab.
I know it's tough. I had a fellowship in Munich. That was tough to have, and then I'm supposed to spend 9 months in 3 years, 3 years appointment. At least I should spend 9 months. I could only get like 30 days maximum in 3 years.
Really?
Then I returned all the funding. They gave me a postdoc and plenty of funding as well, and lab instrumentation use and things like that. But
So,
it's quite demanding, and with the recent government in US, even more so because we are not allowed to take up running labs. It's not easy in two
no, no,
different places.
no, no. But I don't have an option because I want to do cryo-EM and there are no microscopes in Israel. Like there are only two microscopes and they are in the Weizmann Institute and we don't have access to them. So basically, I don't have an option.
Yeah, I
So
understand
I
that.
have to
Yeah.
open the second lab in Germany because I have to do the cryo-EM work.
Great. Sure,
It's
you will do well. You will do well. I mean, the distance, maybe it's like how many hours travel
mm,
Hamburg to.
it's a full day because it's not a direct flight.
Ah, okay.
So I can either take a flight to Berlin, which is about 4 hours, but then like a train to Hamburg, which is 3 hours door to door, something like that. So it sums up, with the travel to the airport, and it sums up to about 12 hours
I'm
travel time.
sure once you set up, it will be things will be easy because with this pandemic it trained all of us to do virtually, so
Yeah.
it might help you to survive through. So
Yeah, but then
John
like
is John. Good morning.
Good morning.
hey,
Looks
how
like Bikash has put a password, so I don't see many participants. Usually, you get a lot of people joining at this hour. I myself had difficulty in logging in, so several complained that there is a password to join. Was it easy, John, to join?
There's always been a password for me, a number.
Oh, okay. For some reason,
Oh
it
no,
asked the
no,
password.
you're right. It's not a number. It's just the
It's just a number, yeah.
code.
Yeah.
Yeah, you're right. Hey, Meytal, how are you?
Hey, how are you?
Good, good.
Yeah.
I just woke up.
Yeah, it's very early for you. Like, it's like every Saturday to wake up at this hour. It's crazy.
I know, I feel bad
Yes.
for John. John, you see, our time is like 9:45. I woke up at 9:15. See my coffee? What's the magic molecule to?
So, Meytal, I'm gonna go and get myself a hot tea, and I'll be back in a few minutes.
Okay.
So let's check your slides. Meanwhile, Meytal,
Yeah.
can you? You should be able to share your screen now. You see at the bottom, yeah, that's working. Now, can you go forward and reverse to see if it's working? Yeah, good. Do
Yeah.
you have any animation you want to test it?
It's a GIF file, so it shouldn't be
Okay,
a
good.
problem.
That's good.
And it's not like terribly important.
Nice. Okay, so can you get your laser pointer?
I don't know if like, do you see?
Yeah, I see the cursor. If you want to use laser pointer, you the left bottom. Yeah, you know the place. Okay, not that one. The second from
second
left.
one.
No, the next.
Oh.
No. Go back to the left bottom corner.
It changed
You see
something.
that there's a crayon like on the left. No. No, one more. Yes.
This one?
Yeah. Click on that. You'll see laser pointer. Yes. You see that? You can change the color of your liking. Okay, it's working. Everything is working. Magda is here.
Magda is here.
Yeah. Magda, you made it. Good morning,
Hey,
Magda. Magda, unmute yourself.
Oh, I need to.
You need to wake up.
Yeah. Why this is? Oh, finally. Yeah.
Good.
Hey, Meytal, how are you? Nice to see you.
Nice to see you. Have activities now in the morning and in the evening.
Oh, yeah. Lots of stuff going on. Yeah.
Yeah, for you, the Eisenberg is like the middle of the day. Like,
yeah, it is not fun. It was fun, but the middle of the day is kind of really difficult because you want to do something on the weekend. For you, it's kind of not so bad because it's at the end of the day.
Yeah, it's the end of the day, but I'm getting so tired. Like last time I got really tired and I don't know, like I'm not sleeping very well. And like 11 at night, I'm like
yeah,
kind of dead. And
yeah.
you know, but you remind me, maybe I should write to Einat, like
yeah, we need to write her. I don't know. She was. It just we need to track her. She likes to disappear.
I'm writing her now.
Yeah. So yeah. So it will nice to see her, and I need to bug Marcus because I don't know. He needs to show up.
Yeah. Why? What did he tell you?
He's picking that one picture which he wants to show, and he's still picking it up and saying, "I tell him you have thousands. Why one picture?" Yeah. Anyways, so
I'm just writing Einat now.
okay, yeah. I just need a couple more minutes too. So. Everything is working, Rams, right?
Yeah, everything's working. We checked it.
All right. Okay. Thank you.
Yeah, so I just wrote her.
Yeah, great.
Yep. Yeah, Saturday morning, like I applaud you for the for these efforts.
Yeah, that was really great that they are doing it.
Yeah.
So
what's the situation there? Like, do you have in Michigan like the lockdown? You have a lockdown kind of, or?
Oh, we have been having a permanent lockdown. has been more or less all the time, and it doesn't feel, when they say, oh, it's a lockdown, doesn't really feel like a lockdown, because
But there are schools. The schools are open. Like
no, everything is online, and this is the difficult part.
yeah, this is the problematic part. I think that there is no school. It's very difficult.
Yeah, I don't know. Do you have school for your kids, or?
No, like my daughter returned to school, you know, in kind of a partial mode, and my son just this week returned but only like, you know, few hours a week, like one day, something like that. And I think they are really missing the activities and the social interactions. So
Yeah, this what I'm missing, and I'm trying not to think that much about that, because it's difficult, and I usually go to Bulgaria, and I missed that year, so it is difficult.
yeah.
Meytal, how old are your kids?
My son is almost 16 and my daughter is almost 10.
Yeah, because I have an 8, a 12, and a 16. And the 12 and the 16 are completely online, and it's okay. But for the 8-year-old, he was completely online, and it was just a disaster.
Yeah.
And so I actually just moved. So the public schools here are online, but the private schools are in person. So I just moved him,
Oh, really?
and he's so happy.
Okay.
I mean, he's so happy. It makes such a difference to their mental state. I mean, it's like he's a different person.
Think about the consequences that the public schools and the private schools have different corona criteria. Like, what does it do? Like, it doesn't feel right? Like,
It's not right. And but actually, the CDC recommended that schools reopen, but it seems to be the teacher unions in the public schools who don't want to. They said they were going to strike.
Yeah, I think this is a general problem. I was in Germany in a sabbatical. Like, we returned in the summer, and I was so upset that we returned to Israel, because in Germany they have the schools open as usual, because they said this is not a reason for the outbreaks. Like, they managed to handle the school, so they didn't even close the school for a day since August.
Wow.
Like, just, you know, normal vacations. And I'm really upset that I brought the kid back to Israel. But I didn't have much choice, but still, like.
Sorry to interrupt. Magda, it
Mhm.
looks like because we had a break and not many are in the attendees, so we may have to delay a little bit, 5 minutes or so, maybe
Oh,
9 to
okay.
5,
Yeah, that will
so,
be good. Yeah,
because we must be catching up.
yeah.
Usually we have plenty of people joining.
How
It's
many
because,
do you have normally?
We have 700, more than 700 participants registered. Depending on the day and speaker, we have like anywhere from about 90 to 150 joining
Oh,
live.
wow, that's a huge difference.
Yep. David Eisenberg had a maximum number of participants, huge crowd, and lots of good discussions.
It also depends, Meytal, on the holidays, because like there are lots of holidays going on all the time. So.
But you still have a seminar next week, and I guess then you take
No. We got two more. We got two more.
2 more.
We are
Okay.
working until at least December 19th.
Okay, wow.
And then we'll start on January 10th. So we give her like a long break. Okay, we got 10 minutes. If you want to take a break, Meytal, if you want to just relax, you can.
Yeah, I'll go and
You can close your video and then relax.
okay. Yeah, I'll just go outside, like, to get some water.
Okay, Meytal, I'll see you in 5 minutes. So
Yeah. Okay.
yeah.
Magda.
Yes.
Do you look at the chat folder? I just posted the information for students and postdocs to be aware of. You can also mention just one line saying that we will be coming with the format so that that would enable them to participate live. So I just posted that. Just take a look at it, a couple of sentences, and then you can mention a sentence or two.
Oh, okay. Yeah, thank you.
Magda, are you there?
Yes, I'm here.
Okay, so I don't know if we can make you host, and then I don't know what I can make you host. So because my internet is a little bit slow, and then it just crashed, and then now it came back. You didn't see that change,
Okay.
right?
Yeah, I'm fine. It's
I'm
just I'm
worried if I
still
make you a
here.
host, I don't know if it works or not. So I just don't want to make any alteration at this last minute.
No, I'm fine. I see
Okay.
everything, so it's really good.
So for some reason, Bkash didn't make it somehow. Okay, we can manage it, no problem.
All right. Yeah, yeah.
So we'll start at 10:05. 5 minutes delay. Okay.
All right. Okay. Thanks.
10:04, I'll give you the bell sound, and then you can get started.
אני נותנת הרצאה עוד דקה. אני אסגור את הדלת. בבקשה.
I see, because you made it. That's good. So Magda, are you there in 30 seconds? Magda?
Yes, I'm
Okay, in
here.
30 seconds, okay.
Yeah, I'm here. So, hey, Meytal, you're here too. So everybody. So do we go ahead and start?
Go ahead. Go ahead.
All right. Before I start with the introduction with the guest speaker today, I really want to thank you everybody for coming to the ZOOMinar series today. And I want to remind to the students and postdoc participants that we will be soon coming with the format for which you will have the opportunity to give presentations on your own work, and this will be sometimes in the spring of 2020. So you just need to stay tuned and watch out for our announcement. What you will need to do is to post a short video on your research findings, which it will be evaluated by a committee, so we can select who will be presenting in the postdoctoral series of seminars. As usual, if you have questions, please drop us an email. And today is my real pleasure to introduce Meytal, Professor Meytal Landau. She received her bachelor's degrees in Hebrew University, Jerusalem. And then she moved on with her master's in neurobiology with an advisor, Professor Nava Zizapel in Tel Aviv University. And after that, she continued with her PhD degree, again in the Tel Aviv University, where she worked with Professor Nir Tal to study protein families using various computational methods. After she finished her PhD degree, she came to the States and joined the laboratory of Professor David Eisenberg at UCLA. This is why I know her. And she studied extensively the structure of amyloid proteins, and in particular, how they interact with various inhibitors. And in 2012, Professor Landau started her own lab in Technion Israel Institute of Technology, which is in Haifa, Israel. And currently, her research focus are microbial amyloids and other functional fibrils. And she's a recipient of many awards. The one which I really want to mention is Margaret Oakley Dayhoff Award from the Biophysical Society. And I really don't want to steal any more of your time, Meytal. I'm so glad to have you here and looking for your seminar.
Thanks a lot, Magda, and thank you for the invitation. Yes, we go back a long time. And I'm happy to present our work on functional fibers, which I think are fascinating systems. And I really hope that by the end of my talk, all of you can appreciate how special they are and different from other protein systems. So The functional fibrils that I'm going to talk about are, some of them, they serve as virulence determinants in microbes. They can be either involved in the structuring of the biofilm of different bacteria and fungi. And some of them can also act as cytotoxin. For example, they are killing our immune cells. So they're being secreted during a bacterial infection to the blood, and they can actually kill the T cells to prevent us from attacking the bacteria. And another set of functional fibrils that I'm going to mention are actually coming from all kingdoms of life. They are antimicrobial peptides that are being secreted from bacteria, from amphibians, from human, and they also form these fibrils. And the motivation for this work is actually started because some of these functional fibrils from microbes were shown to have similar biophysical properties to amyloids. And this was surprising. Well, like, what is the connection? We know the amyloids that are more related to neurodegenerative diseases, as you all probably well know. But we also know today that even in humans, not all amyloids are related to pathology. And also some of these amyloids in humans, maybe they have the original functional role. So nobody knows what's these functional roles, but some of the hypotheses also claim, for example, for amyloid beta and alpha-synuclein, that they have some antibacterial activity. So there is a connection that is growing between antibacterial activity or toxic entities and amyloids from different kingdoms of life. And as you know, that one of these diseases that is super interesting is that there are prion diseases, there are transmissible diseases, and as you well know, the Stanley Prushner dogma of transmissible protein, that it was very, very hard to expect. And you know, as structural biologists, we always wonder what makes a protein having this unique kind of properties that you can digest it and it's surviving your GI tract and reaches the brain and templating other proteins. So you all probably heard about it so many times, so I just will remind you that the first atomic resolution information on the structure of the backbone of the amyloids is coming from short segments. Magda was deeply involved in this project that was conducted by David Eisenberg, and in 2005, the first structure was solved, showing the formation of these tightly mated sheets forming from segments tucked in register, and they are forming this steric zipper configuration that is highly stable and illuminates both the stability of the amyloid fibrils and their self-seeding characteristics. And since then, the Eisenberg lab had determined more than 200 structures of these short segments that presumably recapitulate the backbone of the amyloid fibrils from different amyloid-related proteins. And with time, and a solid-state NMR and the resolution revolution of cryo-EM really picked up. So now we know more and more structures from this amyloids related to neurodegenerative diseases, and the cross-beta conformation still holds, just we see more and more polymorphism. So we definitely know that the Anfinsen paradigm of one sequence, one structure, one function doesn't hold for amyloids. And we also know that probably the same sequence can have different morphologies that might correlate it with different level of toxicity. So, but this is it for related to neurodegenerative diseases. I will mention the connection briefly later, because I want to move to what I'm really working about, which are amyloids that are being secreted by bacteria. And for example, in the biofilm, the amyloids are holding this biofilm together. So for those of you who are not familiar with biofilms, they're agglomeration of bacteria that secretes all kinds of materials outside. It can be DNA, protein, exopolysaccharides, and so on and so forth. And when you have this biofilm, imagine it like a tennis rack with all the bacteria sitting on it. It's very strong, it's very adhesive. It can attach to tissues, to skins, to medical devices, to pipes. And if you have an infection that involved a biofilm, you cannot treat it with antibiotics. You have to surgically remove it. If it's on an implant, you have to take it out. It's highly resistant, it's highly resilient, and it's a huge it's a huge health and environmental problem. And I got interested in these biofilms a few years ago because they secrete amyloids that are often give the stability and adhesive properties of the biofilm. And when we started this work, there was no structural information available whatsoever on microbial amyloids, and we got really curious. So at the beginning, when we worked on these biofilm proteins, we used the Eisenberg approach to identify the backbone of the fibril, because even up to now, there is no structure of a full-length protein that is involved in the biofilm. It's highly challenging to solve this structure. I will talk about it a little bit afterwards, but the only structural information at high resolution that is currently available is coming from these short segments that might represent the backbone of the amyloid, although I'm totally not sure about it for the biofilm, but this is what can be done for high resolution structural information. And we took different amyloids from different biofilm related bacteria, and every time that we solve the structure of these segments from the biofilm associated proteins, we kept on getting the steric zipper. So on one hand, you know, in the structural biologist, it wasn't that exciting because I've seen at the Eisenberg lab many of these structures, but on the other hand, it was the first time that we showed it at atomic resolution in bacteria. So it's kind of a motif that goes all the way from bacteria to human. It's probably very strong in evolution. Some of you may know the hypothesis started by Udi Gazid and then Roland Rick and colleague about amyloids being maybe the prebiotic molecules with RNA, maybe with lipids, and this is definitely a strong motif, so it's not surprising that we see it in bacteria. But in bacteria, we actually understand better about the role of this steric zipper because it's such a stable structure, as we mentioned for the prion protein that can sustain all the GI tract. So it makes perfect sense for the stabilization of the biofilm, you have something as stable as this cross beta configuration. And then we thought, what's the implication? Okay, we found that they have a similar backbone structure between the biofilm-associated amyloids and the neurodegeneration related amyloids. And maybe if they have indeed a similar structure, maybe they can be inhibited by the same compounds. So we collaborated, we did a real world from Jülich in Germany, and they had several compounds that were already in clinical trial for Alzheimer's, and we tested a bunch, and we found 2 compounds that also inhibited the fibril formation of the biofilm-associated protein from enterobacteria, from E. coli and Salmonella. And we tested this inhibition in different ways. Like, for example, here you see the electron microscopy. So we saw that in the presence of the compound, there are no fibril formation. We did a tau-flavin T analysis. We did a CD analysis to see that it prevents the transition into beta structure. But actually my most favorite kind of essay, and I'm only talking details about the details of experiments here. This is the only time that I'm going to talk details, I promise. But I want to mention this specific method that we use, which is simple SDS page gel. And the reason for that is that if you take the fibrils, if you grow the fibrils of these biofilm-associated amyloids and you boil it in 95 degrees in SDS buffer, nothing happens. The fibrils are not breakable. They even continue to grow in this SDS buffer, which is totally crazy, and it's true for other harsh solvents as well. So there are crazy proteins. I call them lab killers. Like if you want to try to use them on a FPLC machine, it's killing all of your columns. It's just destroying everything that it touches because it's so aggregative and so sticky, you cannot get rid of it. But we use this property that it doesn't dissolve in SDS to test the inhibition on a gel. So if you do have more monomers, it's more soluble species, it will run on a gel. And once it forms the fibrils, it doesn't. So this is just another method, but it's also it's showing you how robust, crazily stable these proteins are, and probably related to this steric zipper cross beta conformation. And the nice thing is that these compounds didn't just inhibit the protein, the fibrillation of this amyloid protein, but they also reduce the biofilm biomass in the actual salmonella. So think about it, this is pretty cool that we can repropose drugs from anti-Alzheimer's to anti-biofilm based on the structural similarity that we discovered for this protein. So this was pretty nice. But then we thought about another implication that maybe it's less nice to think about, is that if they have a similar structure, maybe one can cross-seed each other. And Matt Chapman showed it a while back also, and we kind of repeated this, that the seeds of these bacterial fibrils, they nucleated the fibrillation, they enhanced the fibrillation of amyloid beta. So I don't know if this is, something that what we see in vitro, we can translate to in vivo, and Matt Chapman has beautiful work now also on alpha-synuclein and the connection between these bacterial amyloids and human neurodegenerative diseases. And there are a lot of correlative evidence showing that if you take mice models of Alzheimer's and you feed them with bacteria or the bacterial amyloids, you have enhanced symptoms. But we definitely need more molecular level understanding of this, what we call the gut-brain axis. What's the connection between aggressive infections and maybe even our microbiomes and neurodegenerative diseases? And is it that far-fetched to think that if a protein from a cow can actually template and cause a neurodegenerative diseases, is it that far-fetched to think that maybe something from bacteria can do it as well? So, and bacteria can affect the neurodegenerative diseases in several ways. There are this direct cross-seeding between the amyloids. Maybe it causes inflammation leading to neuroinflammation and then to neurodegenerative diseases. Maybe the microbes themselves somehow activate the cleavage of amyloid beta, and then you have a lot of amyloid beta around when you are facing some kind of an aggressive infection, and maybe 30 years later, it causes Alzheimer's. There are some evidence for deposits, like joint deposits of some bacteria inside the amyloid beta plaques in the brain. So there are a lot of connections, but we do not totally understand what is going on. So it definitely deserves more research, to say the least. But I want to leave you with this, not this, nice thought, and just move on to a different family that I want to talk about. So I want to talk about this bacteria. This is called Staphylococcus aureus. It's kind of a nasty bacteria. It causes a lot of health issues and infections that also result in death. And some of the species, strains, are resistant to all kinds of antibiotics, like the MRSA strains. And this bacteria is secreting a bunch of amyloid peptides called phenol-soluble modulins. And several years ago, the lab of Blaise Boulez, also from Michigan, they also showed that some of these phenol-soluble modulins, or PSMs in short, they form these amyloid fibrils. And we started to investigate them structurally. And we started with PSM alpha 1 and alpha 4 that were shown to be involved in the biofilm of the staph aureus. And Nir in the lab showed that they form cross-beta amyloid fibrils by fibril diffraction and other methods. And when we solved the structure of the backbone, we got a steric zipper. So this is just another example of biofilm-related amyloid that is forming the canonical cross-beta configuration. But then we tried another peptide of this family, PSM alpha 3, and at the time we weren't even sure if it forms amyloid fibrils or not. So Einav in the lab, who is now actually a postdoc with David Eisenberg, she showed that it forms nice fibrils. they bind tau-flavin T, and they form these beautiful crystals, which look like normal amyloid. Like it looks like all the amyloid crystals that we got before. And I was really surprised because we crystallize the full-length peptide, which is 22 residues, and we never crystallize something as long as at the Eisenberg lab. Like the maximum was 8 amino acid. Usually it was 6 or 7 amino acid, but not longer than that. And I kept on sending these crystals to mass spec because I couldn't believe that it crystallized, but it did, and it did the full length. And as David Eisenberg told me once, long-lived crystallography, because it revealed like how is it possible that we form the fibrils. And when we saw the fibrils, we saw something really interesting. We saw the canonical configuration of this single molecule stacked perpendicular to the fibril axis and mated sheets. But the individual molecules were formed out of alpha helices and not beta strands. So if you just compare what we call the cross alpha and the cross beta, you can see a very similar quaternary structure, but it's composed of a different secondary structure. And I think this is really fascinating that you can see this architecture made out of different secondary structure. And even the idea that you can form alpha helical sheets is something that is relatively interesting and rare. Of course, you don't have all the hydrogen bonds between the strands, but you do have some salt bridges and other form of interactions that stabilize the entire thing, and especially the hydrophobic core between these sheets. And I call them sheets. I will show you later why. And then comes the question, what are these cross alpha fibrils are doing? Like, are they functional? Like, why are they formed? Like, do they have some purpose? And we knew that PSM alpha 3 is the most toxic member of the family. So if you incubate T cells with these peptides, like 5 micromolars, after 5 minutes of incubations, you just get a graveyard of T cells. It's just a killer. It's killing the cell. So the question is, what's the connection between the fibril formation and this toxicity assay? And this is not a trivial question to answer. And we did a lot of experiments. I would not, get into the details. I'll just explain the line of thought. What we did is extensive mutagenesis analysis, and we look on the fibril formation, the properties of the fibrils in different secondary structures, and correlate this with toxicities. Just to summarize the results, we came up with a conclusion that fibrillation is important for toxicity. It's not sufficient, because if you lose the positively charged, you also lose toxicity, but it also enhances toxicity. So if you don't have fibrillation, you reduce toxicity tremendously. And then we kept on monitoring on what happens to the peptides when encounter cells, and we kind of observe this aggregation, and the cells just suck in the peptide. So there is an attraction between the cell and the peptide, and you can see that the peptide is co-localized with membrane particles. And so we used this microscopy, and we also collaborated with Razi and Linek from Ben-Gurion University to analyze the membrane, the membrane interactions between these peptides and the membrane. And we came up to this conclusion that the peptide and the membrane, they affect each other. It's not just the membrane is sitting there waiting to be affected by the peptide, but actually by touching the lipids, you have enhanced aggregation, and they are probably, this is the model that we have, they probably co-aggregate together. So the peptide touches the lipid, there is enhanced aggregation and leading to toxicity. So if you have only fibrils in your solution, you have robust fibrillation, probably it will not be toxic, because you need these dynamic interactions. You need the ability to form fibrils, but you also need the soluble species to touch the lipid to enhance fibrillation and doing all together. So I think maybe this concept of thinking of an amyloid toxic entity, such as oligomers or fibrils, maybe we should revisit this idea and not think about it as a toxic entity. We should perhaps look at it as a toxic process, something that is happening. So it's not that the fibrils are toxic, but the ability to form fibrils is necessary in order to kill the cell. So this is what our indication for this specific peptide from staph aureus, this bacterial peptide. I don't know how it's correlated to neurodegenerative related amyloids, but definitely for alpha-synuclein, there is a lot of work that is showing that the interaction with the membrane and the alpha helical conformation of alpha-synuclein. Some members of the crowd also know a lot about that, and I think this coagulation with the membrane is something that is definitely important, and how come maybe helicity also has something to do with this toxic activity. So the plan for us to get more information about this toxic process is actually visualize the process, and what I'm planning to do next is to use correlative cryogenic light and electron microscopy, cryoclem, in order to actually visualize this toxic process. So just to summarize, think about this crazy family of PSMs that this bacteria secretes. Like by secreting these two peptides, alpha-1 and alpha-4, you have biofilm structuring. Alpha-3 is causing cytotoxic activity and is killing our immune system. And these peptides are also being cleaved in vivo to active derivatives, and some of them that they didn't mention, some of them, they have antibacterial activity, and they also form kind of unique amyloid-like conformation. They're beta rich, but they're not amyloid per se. So by just secreting 3 peptides, this staph aureus gets a lot of activity done. And it's not surprising that the highly virulent species of staph aureus are secreting like 60% of the protein mass that they secrete outside our PSM, because this is really an amazing family. And we haven't even begun to look on the other members of the family. So the question now comes is this cross alpha configuration, is this something that only happened once in this specific bacterial protein peptide, or it's something more canonical, it's something that happens more in nature? So we looked for more toxic peptides that form fibrils, and we found that an antimicrobial peptide being secreted by an amphibian is actually also forming the cross alpha configuration. So we solved the crystal structure of this peptide, and you can see another cross alpha, but look at something really interesting. The sheets, I don't know how to call them. I'm doing like like that because we are not used to thinking about helical sheets, but they are in a sense. And you can see that the helices are oriented in an anti-parallel manner. And in the bacterial one, it was parallel. So you have also in these helical sheets, you have anti-parallel and parallel sheets, which I really think it's amazing how they can recapitulate the properties of a beta sheet. And in terms of fibrillation, so these peptides form fibrils, but if you use low concentration, you can barely see fibrils. But if you use the same concentration, about 4 micromolar, and you add the bacterial cells, then you have robust fibrillation even in very low peptide concentration. So again, I think that the membranes are inducing fibrillation, and this fibrillation is eventually killing the cell. So this is kind of a cohort process between the lipids and the peptides to actually cause toxicity. And what was really amazing about this peptide is that it has a chameleon property. So I won't drag you all the biophysical assays, but basically, when you look on this peptide in the presence of cells or lipids, you can see a cross alpha configuration, and you can see much more helicity. But if you don't have lipids or cells, and especially if you induce heat, you can see the formation of cross beta fibrils. So it's the same sequence, no sequence modification. It can form both cross beta and cross alpha fibrils. And it's like our model that it might be related to regulation. So if it's being secreted on this frog skin, it's not a frog, but whatever, like it's a different kind of an amphibian, but let's call it a frog. So if it's being secreted on the frog skin and there is no bacteria around and it's sitting in the sun and it's hot, So it's going into cross-beta fibrils, which are not as toxic and are not damaging the frog cells themselves. But once it sees bacterial cells, there is a transition into cross-alpha fibrils that are killing the bacteria. So it's a matter of regulation, and let's be active only when it needed to be active. Only when we have bacterial cells around, only then there is a structural switch and activity is enhanced. And we have another interesting question: if this kind of change is happening in the solid form, is it possible to switch to cross-beta to cross-alpha in the fibril form itself, or it has to go through monomers? And I'm not sure, but it's not that far-fetched to think about it, because it's the same orientation there. The molecules are situated perpendicular to the fibril axis, and you have mated sheets. So the cross-alpha and the cross-beta have the same architecture exactly. So if you just change the secondary structure, it can rearrange, but it doesn't need to do all kind of massive rearrangement. It can just stay the same. Just the distances are going to be different. So maybe this is not that far-fetched of an idea to see this secondary structure switch in the fibril form. And when we look on stability of this fibril, so if we look on canonical cross-beta, and you see it a lot for cross-beta, they are highly stable also in elevated temperature. The cross-alpha is not stable, so this is definitely a less stable configuration than cross-beta. And when you look on this amphibian, you see that it's still very stable, but we know that high temperature are leading to more cross-beta configuration. So probably this stability is coming from the formation of this cross-beta fibrils. So this is about the frog. And then we started thinking, okay, there is a lot about this connection between antibacterial activity and amyloid formation. And we know that many antibacterial peptides, they form amyloid structures. We know that amyloids, canonical amyloids like amyloid beta and alpha-synuclein, have antibacterial activity. So this connection is coming from both sides. So we wanted to look at more antimicrobial peptides. And Itzik and the lab found that a derivative of LL37, which is some of you know this peptide very well, and Rams, we talked about it like several years ago. But so this LL37 itself forms fibrils, but not that robust. But this fragment, which is the active core of LL37, it's a 13 residue peptide, and it's the minimal length that has antiviral activity and all kind of sort of different spectrum of antimicrobial activity. So we solved the crystal structure, and it's an amazing structure. It forms from this highly dense helices, and the basic unit is actually 4 helical bundle with a hydrophobic core, like the ones that are in green or in purple, that are 4 helical bundles. They have a hydrophobic core, and the connection between these 4 helical bundles is based on more polar and electrostatic interactions. But jointly together, they form this highly dense packed helices. And on the side, you can see that this peptide also forms fibrils in solution. There are ribbon-like white fibrils that are contacting the bacterial cells. And then the question again arises, is this self-assembly important for activity? So we have some indications. If you look on the surface of these fibrils, and you color it by hydrophilic to hydrophobic coloring scheme. So what you see is like zigzag belts of hydrophobic and positively charged. So these hydrophilic areas here are positive charges. So you have belts of hydrophobic and positively charged surfaces. And it makes perfect sense that it's interacting with the negatively charged bacterial membrane. So the surface kind of suggests to you that it might form this interaction with the membrane. And Itzik also conducted a bunch of mutations, and without going through the details, but we did correlate the ability to have self-assembly with activity. So we design the mutation based on the structure, and if we damage something that is very important for the packing, we don't have fiber formation and we lost activity. And if we mutated something that is not as that doesn't form too many contacts in the structure, it still forms these fibrils and it's still active. So the conclusion is that self-assembly is important for antibacterial activity, although I cannot swear that the exact configuration that we see in the crystal structure is indeed the only one that is possible. And these fibrils are also very thermostable. They can, at 80 degrees, you can start seeing this little bit of disassembly, and you know that these ribbon-like fibrils are actually composed probably of many other fibrils that kind of associate together. So it's a very unique kind of fibrillation form. And we definitely look forward to seeing more antimicrobial peptides that form these beautiful fibrils. So to summarize this talk, I showed you a lot of bunch of really different things. I hope that I didn't confuse you completely, because I've shown you biofilm-related amyloids that have similar structure to cross beta. They are being inhibited by same compounds as amyloid beta. I showed you that a toxic peptide from bacteria is forming cross-alpha amyloid peptides, and that probably the co-aggregation and co-formulation with the membrane is important for toxicity. And I showed you that an antimicrobial peptide from an amphibian also from this cross-alpha configuration, and fibrillation is again happening on the membrane, probably leading to toxicity. So I don't know if the antimicrobial peptides have similar mechanism as the toxic peptides that are killing human cells, but both of them are cross-alpha. Probably there are some differences and there are some similarities, and we have to know more about the mechanism. And finally, I showed you that we have antimicrobial fibril also from a human-derived peptide that is not amyloid by any means. If I said amyloid, I was mistaken. So it forms a very dense fibrils, but it's not an amyloid. So we are kind of extending this amyloid fibrillation world to functional fibrils in general. And the implication is that we want to think about new antimicrobial drugs. So if it's compounds that, for example, target the bacterial toxic peptide, so maybe something that will prevent fibrillation of this bacterial toxic peptide PSM alpha 3, you can reduce the aggressiveness of the infections without killing the bacteria and maybe inducing less resistance, but just attacking the weapons or the virulence determinants of the bacteria. And you can also use the amyloids themselves for new antibacterial drugs with this added value that you have peptides that are highly stable under high temperature. Maybe we can design some that will be resistant to proteolytic degradation, that they have a very good bioavailability compared to other antimicrobial peptides. So this is one direction. I think that we can think about materials for novel technology using this highly stable and unique configuration. I think that there is a lot to investigate about how microbes communicate between themselves using these microbial amyloids. And definitely there is interactions between the microbes and the host that are somehow related to amyloids, and we need to understand them further. And what's the connection to neurodegenerative diseases or to human diseases and conditions in general, because we know that the microbiome is highly influential and we need to understand this amyloid component in that. So next for me is that I do want to understand the toxic mechanism better, if it's antibacterial or anti-human cells. I want to understand the formation of mixed fibrils. And for all of this, I need better visualization method, as I mentioned. I want to use now cryo-EM to solve the fibril structure, together with lipids, something that we cannot do now with crystallography. I want to see different polymorphs in different environments to get some, better sense of what happens in more native environments. And for this, I spent the last year during a sabbatical in Hamburg, in Germany, thinking that it deficits the German synchrotron, which they have a building, they formed a building called CSSB, which is a collaboration between 9 research institutes in Germany, and they are focused on infectious diseases, on structure and system biology, so it was the perfect location. And I was affiliated with EMBL, working with Matthias Willmans, and also with Kai Grunwald, who hosted me, and we have a joint postdoc now, Robert, who is being funded by actually the city of Hamburg, and he's starting to do all this cryo-EM on these functional fibers. And we collaborate with Caroline Searing. She did her PhD with Ronald Rick, and she's now heading the facility of the cryo-EM. And Roland, who is heading the light microscopy facilities, is now moved, but we want to work together in order to understand the mechanism of these functional fibers. And this is my lab back in Israel, who did all of the work that I showed you. All the work was done in Israel with collaboration with synchrotrons. Obviously, we traveled a lot to Petra and to ESRF, and with the help of microscopy centers, the Technion and other places in Israel. And since we cannot do cryo-EM in Israel because we don't have the proper microscope, we are kind of relying on the microscopes in Germany and this collaboration with Hamburg. So we thought, okay, let's spend our time and kind of a short release and the lockdown in Israel, because the lockdown was really severe in Israel for a very long time. But we kind of took a break, and this is from a couple of weeks ago that we went actually swimming. So I guess that you were also envious at the weather. This was just like, you know, a couple of weeks, and the degree, like it's 28 degrees Celsius, this water, and you can swim in it and it's not cold at all. So there are some advantage to the weather in Israel. Otherwise, I don't know what to tell you. But, and so thanks for the funding and my beautiful lab and the students, and thank you for your attention.
Thank you, Meytal. It was really great. And it was so exciting to hear all the new stuff which you have been doing. There are questions on the Q&A, and usually we let the participants ask the questions themselves. So Bikash, do you mind letting them go in, or Rams? I just prefer the participants, the audience, to ask their questions.
Because are you there?
Okay. I'm doing that.
Okay, good.
Yeah, Lee, please go ahead and ask your question.
Hey, Meytal. This is Chuyi. I'm always a great fan of your cross-alpha structures. I think, if I understand it correctly, those helices stack 3-dimensionally in microcrystals, and they are like 4 residues per run helices, so they have like 4 interfaces. So how to determine which interface is involved in fiber stacking, which interface is involved in protofilament association, and which interface is exposed to water? I think this is a very important information for you to understanding the interaction with membrane. And also, do you have any experiments to prove this assignment of this stacking?
Yeah, we don't have any, I couldn't conceive of any any experiments to actually prove that. And you're totally right, everything is coming from the crystal contacts, but it's not totally. First of all, let me just say that the cross alpha configuration is not just in the crystals, because we did diffraction of the fibrils. Not in crystals, just, you know, fibrils that we grew in water, and we see a cross alpha diffraction. It means that we see the cross as in the cross beta, but the distances are complementary to the exact distances that we see in the crystal structure. So we know that what we see in the crystal structure is correlated to what we see of fibrils growing in solution. So this is a very important aspect, that it's not just a crystal contact artifact, it's something that is really happening. And it's a good question, how do we define a protein fiber? So for me, in the crystal structure, what we see is actually these mated sheets that have this hydrophobic interface. So it's very obvious to me that the sheets that are being held together by this hydrophobic interface is kind of robust, it's strong, it's stable. So probably the protofilament that you want to talk about is at least 2 mated sheets. But if you think about, if you look on the crystal context, so you have hydrophobic interfaces, hydrophilic interfaces, hydrophobic interfaces, like they're alternating like this. And between the hydrophilic interfaces, there is a lot of water molecules. So obviously, I would take at least like this dry interface as the protofilament. But the thing is that so on this lateral kind of configuration, it's very simple that it's the dry interface. But on the other dimension, they form kind of rows of mated sheets. So they don't have a lot of water molecules between them. So I don't know how many actual rows like that are stuck together in a protofilament. We can calculate this, based on the fibrils that we see in TEM, but there are so many polymorphisms. So I guess that every fiber is composed of different number of these rows of helices in this direction. If this is the fibril axis and this is the hydrophobic interface, I don't know how much we have in this direction.
Is there going to be only one row, or they also have like multiple protofilaments? They were associated like in a 2-dimensional way.
Yeah, just by looking on the morphologies that we get by the negative staining and now with cryo-EM, they're very different from each other. Some of these things are very straight, and some of them you can see that they're composed of protofilaments associating like this, and every time it's a little bit different.
Thanks. Really look forward for your cryo-EM structure of cross alpha.
Yeah. Okay. Should I read the next question, or from Christian? Do you want to ask, or should I read it?
I can ask it.
Okay.
So, given your alpha helical amyloid structure, I would expect that the anti-beta amyloid compounds that you were using have less effect.
They don't work on the cross alpha, we checked.
They did not work.
Yeah, they don't. Okay. This was
Okay.
our control when we published when we published them. They work only on the cross beta, but not on the cross alpha.
And another question, I mean, A beta, for example, in solution has also some alpha helical conformation. So in principle, it could also potentially form such alpha helix amyloids. So do you have any idea what decides whether it then forms helical versus beta sheet amyloids?
Yeah, there is a lot of work on amyloid beta and especially on alpha-synuclein, that they have these alpha helical intermediates, and some of this work is saying that this helical the helical configuration is intermediate to fibril formation, and if we think about that toxicity is coming from the intermediate, so you can see a connection between helicity and toxicity. But in most human amyloids, like amyloid beta and alpha-synuclein, these are just the intermediates, and the final fibrils are cross beta. And this is also true for the amphibian peptide. So if you don't have lipids and you have fibrillation, it will eventually go to cross beta unless you have lipids around. And I don't know, even for alpha-synuclein, I think if you have lipids around, you also kind of stabilize the alpha configuration. So, but the difference is that in the bacterial peptide, the final fibril is helical, and all the other amyloids, the final fibril, the most stable one, is cross beta. So I think it's everything is about kinetics eventually, and thermodynamics. So.
Okay.
Does it answer your question?
Well, I mean, of course, for example, for alpha-synuclein, when it's helix associated, it is alpha helical, but it's more the monomeric state, and normally it doesn't form helical aggregates, and that was my question more or
Yes,
less.
alpha-synuclein does have oligomers that are alpha. Not just the monomers, also the intermediate oligomers, like work at the Dobson lab and Thomas Knowles and other labs definitely that show that. Like John might know better, but alpha-synuclein is definitely alpha also in the aggregated form.
Well, I mean, the published structure from the Dobson lab on the only oligomer that I know published in Science, I think in 2015 or so, has no alpha helical conformation.
Yeah, but they had later work, so I'm not sure how much of it is already published and how much of it I heard.
Maybe you could just send me the paper you're referring to. I would be very happy.
Okay, but I'm I don't remember if it's. I'm definitely certain that they have it. I just don't remember if it's from publication or from conferences. That like later work. But I will find, if I can find it, I'll send it to you.
Great, thanks.
I think we have a question from Long Cheng, but there are anonymous attendee questions, and there is an email attached to a question, Meytal. Do you mind? Let's, Long Cheng, why don't you go ahead and answer, and after that, Meytal, you can check the last two questions on the chat box, on the Q&A box.
Yeah. Hello.
Hi.
Hi. Fantastic talk. Thank you so much. And actually, my one question is, in terms of the neurodegeneration, actually, people looking forward to find strong and clear evidence of where the presence of the bacteria in the brain or virus, but kind of challenging. So my question is, in the neurodegenerative disease in the brain, what is the potential factors can regulate this transition, like structure from alpha to beta and the lipid association? And another question is, look like this structure transformation and the lipid association get involved multiple function like immune response and also toxic, but in the neuron, so this the membrane, I will think about like some fiber will bind to like a receptor like a TLR to triggering immune response. Some like alpha-synuclein can bind to the different organelles membrane like mitochondria or autophagy zone. And so my question is, what's your thought about the roles of like a receptor, pH, environment, different organelles membrane, lipid protein? What's the role of this structural transformation and their functional role? Thank you.
So you mean like in receptor activation, not just the membrane interactions?
Yeah, so the thing is this peptide will be preferred to bind to the receptor on the lipid membrane, or they directly bind to the lipid, or in different organelle like mitochondria, and they bind to mitochondria might change mitochondrial stress, cause cell toxic. But if they bind to the cell membrane receptor, maybe they mediate immune response. So do you think any selective mechanism for different function mediated by receptor, pH, or different organelle? Thank you.
So the toxicity itself, I don't think it's mediated by receptors or proteins, because if you synthesize the same peptide with D amino acids, it's as toxic, and it forms also cross alpha fibers. We did like this, and also the lab of Sam Gelman also did this, like with the D amino acid, and you have the same toxicity mechanism. So because receptors they have a stereospecificity, so we don't think the toxicity is related to receptor activation. And also the process is super fast, so it's probably leaking, like it's a lytic process that is killing the membrane. But this peptide definitely activate receptors, as you said, like in the immune system. But actually, like we kind of looked at that in collaboration with a group in the Netherlands. And I think, like I'm saying it with a lot of cautions, but I think that the receptor activation is not mediated by the fibril form by soluble species, and their concentrations that are needed to activate the receptors are much, much lower. So I think that the mechanism of receptor activation by amyloids and also this bacterial protein is very, very different than the mechanism of toxicity. There is a correlation, like one thing that we noticed is the fragment from LL37, the human LL37, and the PSM alpha 3, they are very similar in sequence. It's like scarily similar, and they both form helical fibrils. So we did think about something like molecular recognition mechanism, maybe hijacking, maybe the bacterial peptide is kind of hijacking the immunomodulation roles of LL37. So, you know, it's just not coincidental that they have some similar sequences. So there's something there, but I don't think that it's the fibrils that is active. So I don't exactly know what's going on. So this is the thing. I think it's a different mechanism.
Yeah, thank you. Thank you.
Okay. So Magda, should I take from the Q&A, like can one predict from sequence whenever you can get alpha helical amyloid fibril structure? So
if you can go with the anonymous question, because the one before, which is the participant, is just we let it's on the queue. So why don't you answer the anonymous one?
What do you mean, the one from at Chris? There is no evidence that that amyloid beta is alpha helical conformation.
Yes, that's the anonymous. Yeah.
Well, as far as I remember, there are several papers that do talk about intermediates with helical conformation of amyloid beta. There is no structure that shows helical conformation of amyloid beta, but there are definitely evidence of intermediates. I need to find the papers. I honestly don't remember. And for alpha-synuclein, I'm totally sure that there are indications of helical intermediates. I will have to find the papers and send it to the forum. So,
Meytal, I can give you some evidence here. So the tendency to form alpha helical structure depends on the conditions as well. So, for example, Adbax published A beta being disordered structure. That's one sample, and you don't see solution NMR being detected in any aggregated species. So disordered structure doesn't mean that's given for all possible samples. So Teplo has shown helical intermediates. We have shown 310 helical containing disordered alpha helical structure for A beta 1 to 40. And people have shown intermediate structures for other papers also available. Yeah.
Yeah. So the Teplo lab, yeah, the Teplo showed.
Right.
Right. Yeah. Yeah, I should have remembered this.
So yeah, I think we like the next person on the queue, Meytal. We need to.
For me, what I just see, maybe I don't see them all.
Yeah, the question.
It's on the top, like, but I don't know why we can't hear it.
I mean, is it my question? Then I could
yeah. It's for your question. Yeah, you can hear it. Yeah, now it's fine.
Sorry, it's Markus Zweckstetter from the DZNE in Germany. Hi, Meytal. So actually, I really loved your talk, I should say, and I also loved your image with the tennis racket. But I also have a question. So in terms of, I mean, for synuclein, of course, you can predict, and it's alpha helical, and then one is surprised that it forms beta structure. So how is it for these peptides? Now, I mean, if you do the sequence prediction with Agadir or whatever, are these alpha helical or are they beta, which then form a cross alpha helical structure?
Yeah, they're like the PSMs. They are predicted to be helical, and there are amphipathic helices. But it's kind of tricky because PSM alpha, the entire like alpha PSMs are predicted to be helical by secondary structure analysis, and CD measurements in solution does show the helical content. But PSM alpha 3, and they have very similar predictions, but PSM alpha 3 also, when it forms fibrils, it remains helical. But PSM alpha 1 and alpha 4, although they are predicted to be helical, with time you see the transition into cross beta structure. But it's true, as you said, for alpha synuclein, parts of the, I think the N-terminal region is also predicted to be helical, and eventually you see the transition. So I think for many of the amyloid, they are predicted to be even helical or disordered, but they have this transition. And alpha 3 is unique because it remains helical. But we kind of developed in the lab now kind of a computational pipeline, and we screen a lot of antimicrobial peptides and toxic peptides. And many of them are predicted to be helical, but we actually kind of can pinpoint some of them that has what we call chameleon properties. So in secondary structure prediction, you can see both the helical prediction and some propensity to beta. And I think we are kind of right in this prediction because they can be a chameleon secondary structure switch. So it's pretty cool, actually.
Can I ask a question?
Could I follow up one?
Go ahead. Sorry.
So for the PMS alpha 3, right? I mean, there are toxins which make, let's say, helical transmembrane pores. So do you think that the toxicity could be related to this, that the PSM alpha 3 inserts as a helix in the membrane and that causes toxicity leakage?
To be honest, I don't think it's pores. I think it's like, I don't know. Until we'll do this visualization that I really want to do with cryoclem, I wouldn't be able to tell. But what I envision in my head is that it's kind of, you know, the soluble species and some kind of small fibrils are touching the lipids, and then they're kind of taking the lipids out. It's like kind of chewing the membrane. It's kind of, the lipids are going into this massive aggregate with the peptide, and so it's not porous in like helical with this thing. It's just kind of chewing. I don't know how to. I don't think it's so fast, you know, like the cell is just like ruptures. It's not that you have leakage from the cell. It's just like you know, splashed on the surface, like, you know, exploding. So.
But then would you want to use these peptides really as a drug delivery? I mean, isn't that a bit dangerous?
I think they are too toxic to be used as drug deliverers. Maybe the ones with the antibacterial properties and maybe less toxicity. I think these peptides are actually drug targets, because if you think about it, if we think that fibrillation is important for toxicity, and these are virulence determinants that are killing our immune cells. So if you find fibrillation inhibitors for cross alpha, then you can use them as antimicrobial drugs, like antivirulence drugs, no, not, you know, killing the bacteria.
Thank you.
So Meytal, the mechanism that you mentioned is also similar for other amyloid peptides like A-beta as well, IAPP as well. They rope up the lipids, they pull off the lipids when they form detergent-like membrane disruptions. So they cop it and then disrupt the membrane structure. My question is, you mentioned about alpha helical structures to be more formed in the presence of bacteria, and then beta sheet in the absence of bacteria, right? So you also worked on interesting molecule LL37. LL37 as a monomer, unstructured, and forms alpha helical oligomers in solution as well as in membrane. So do you think, just touching upon Griesinger's, the intermediate alpha helix going to alpha helical fibers, that's maybe the case for alpha helical structures of LL37. So do you think LL37 oligomers are more functional than alpha helical fibers? Do you have any evidence for activity for the oligomer versus end product fibers?
Yeah, I don't know, because think about like, when we think about oligomers in the amyloid world, and we think about kind of intermediates that have to change their secondary structure, right? Like if we think about some intermediates that are alpha, but eventually they are going into beta. But with the helical ones, LL37 is more complicated because LL37 is not an amyloid and you don't have continuous sheets. With the cross alpha, if you think about it, what is an oligomer actually? Oligomer is just a short fibrils because there is no secondary structure switch. So, you know, it's kind of building, like the oligomers will be, you know, several helices bundled together and then they continue, right? So it's totally on path. So it's short fibrils rather than intermediates. And for L37, I think it's much more complicated. So the full-length L37, there is crystal structures from
NMR structures.
From there are also crystal structures.
Yes.
From Cornelius is from Copenhagen.
Okay.
And there are crystal structures, and you can see kind of something that looks like an oligomers, but it's not as densely packed. But the core, this 13 residue, which is also functional, forms these very, very dense structures. So, but I don't know, this is not an amyloid, and it's not continuous sheets, and mutations tell us that self-assembly is important. But to be honest, I'm really not sure that what we see in the crystal structure in terms of fibril is the actual, what is killing the cell. So with this respect, I think there is a lot more that we have to figure out.
So in that context, alpha helical structures as well as LL37 and other AMPs, antimicrobial peptides you mentioned about, do they all show selectivities? Many of the peptides are selective to gram-negative versus gram-positive. And then the membrane structures are very different. In gram-negative, you have huge outer layer, outer membrane. So there you LPS and outer membrane proteins as well. So do you see these your structures to be selective towards one versus other bacteria?
So I think so, but I think the selectivity is not necessarily coming from what they target, but what they respond to. So let's say that the LPS can affect the peptide, and the effect is changing the toxicity. Sure. So the LPS and the lipids and everything is not just sitting and waiting. They're actually being involved, and they're determining their fate.
Sure.
So,
but to start with, they need to be attracted, right? So this electrostatic interaction gets attracted towards LPS as opposed to anionic membrane.
Yes, yeah, definitely.
I mean, cationic membrane, sorry. Yeah, very nice. Thank you.
Meytal, I also have a question. Have you tried to do the molecular dynamic simulation of the transition to see it real time between cross alpha fibrils and cross beta fibrils?
We haven't tried. Like, it's something that I'm thinking on, but we we are not a computational lab. Maybe one of my students is more computational, will try to do this. This is very, very hard to simulate because they don't want to be, you give it a simulation, so maybe the helix will break, but I'm not sure that the simulation can reform it into beta. I think it's too much to ask of the simulation. But we need to try in order to really say, maybe I don't have enough faith,
I understand. But the thing is, because I got intrigued. It's really amazing that they can transition from one state to the others, and since you have the two kind of end states, you may be something, because you can kind of give you an idea as what you can do experimentally and what is affected. For example, do you find a residue which is, if you mutate them, you prevent that transition from one state to the other? You've talked about abolishing the formation of one or the other, but do you have you thought of doing that? It's again, I still, the way I gather it, I think it's these alpha helical cross alpha fibrils looks like they're quite unique for the bacteria, and for that they should be something which makes them to
also
form.
in the frog, we solved the structure of the alpha in the frog, so it's also in eukaryotes that you can see this cross alpha configuration. And I think this is a good system to test the mutations that you were mentioning. I didn't see mutations that can change the chameleon property, but in PSM alpha 3, the bacterial one, we have mutations that shift. So it's a single point mutation can shift more into beta. So you can see, you always see a mixture, but definitely mutation are shifting from one place to one conformation to another. And it's pretty crazy because with alpha 3, if you add lipids that mimic eukaryotic membrane, it's actually making the helices more stable, it's shifting the mixture, like it's completely helix. But if you add lipids that mimic bacterial membrane, which is more negatively charged, then you actually have a shift to beta. So
yeah, it's
a very complicated system because you don't know what is actually touching what.
Yeah.
You know, in what conformation.
Yeah, that's really cool. And I also want to ask you a question about the biofilms formation, because again, biofilms, you know, they are all made of amyloid fibrils and stuff like that, but amyloid fibrils are kind of loose. They are not glued together. I mean, it's just, they won't stick so permanently. Do you know if they're like, just this tells how ignorant I am. Are there any additional things which kind of, if you want, they glue them together, like just the glues in between kind of pointers? And what is the structures of these? Do you
in the biofilm think that there is so much other compounds involved, and it was shown that DNA really affects the fibril formation and the polymorphism and activity, and actually DNA can make some of this CSGA, like match up on CSGA, actually toxic. So DNA can affect what happens in the biofilm, but there are many, you know, amyloids from bacteria and fungi that are adhesive. So they're actually called adhesins or hydrophobins, and they're kind of sticky to the membrane on top of making the amyloids. So it's either like inherent property of this bacterial amyloids that are different from the human ones, but also interactions with other components in the biofilm, like DNA, for example.
So the DNAs can be that glue, which kind of keeps them together, not to be so loose and stuff.
Yes, and you know, it's like you have the exopolysaccharide, you have the cellulose. It's a very different, you know, you have all these things outside that, you know, glue everything together. So it's very different than the environment that you have in the brain.
And the other thing is, which I want to, this is my last question, I just don't want to occupy. But what you've been mentioning, that when they shift into beta structures, they become less toxic to the bacteria. But yet the human amyloids, they are actually toxic to the bacteria. Have you kind of looked into to see what it makes one toxic, the other one non-toxic? They're both cross beta fibrils. Do you see where I'm going?
Yeah, I see, and I don't know, and I want to, you know, advocate, but you know, I'm kind of, you know, like this hypothesis that is the intermediates that are more helical are the ones that are killing the bacteria, not the final cross beta configuration. But I'm not sure, because we see all this like this chameleon sequences that are also cross beta, but they are antibacterial sequences. And I'm not totally sure that the cross beta is kind of a reservoir, and maybe it's also have some activity on its own. Like I want to think, and it makes more sense that the helical intermediates also in amyloid beta and alpha-synucleins are the ones that are killing the bacterial cells. And the cross beta is actually detoxifying, like with human cells, but I don't know. Like we need a plan for that. Like,
yeah, that's because that's kind of intriguing. And the reason why I think what I have in mind, but I glanced it long time, that they are actually indeed the like the fibrillar structures which are killing the bacteria, but I just need to go back and check it. It was quite some time when I was looking at it.
These hypotheses keep on, you know, changing and updating, and
yeah, that's really
Meytal,
cool.
I have a question to ask. Do you see lipids at all in your structures? I know you are growing in solution. Did you ever try with the lipids? Even free lipids, little bit concentration of lipids?
Yeah, so into crystallization, it's a problem to add the lipids. Like we never really tried, but we see MPD, which is kind of a solvent that has some you know, it has some kind of a hydrophobic lipidish properties. So we see it stacked, like, you know, among the fibers. So I can imagine that the MPD is like pushing the cross alpha too.
What happens if we take the alpha helical structures or beta sheet structure? Doesn't matter. Add lipids to it. Do you see they're stable in the presence of
So
lipids?
we didn't solve a high resolution structure, but this is what I want to do now with the cryo-EM to, you know.
I'm talking about the solution structures. If you take those structures and add lipids to them, are they stable?
You mean like, yes, like when we mixed
Catalyst to the lipids, yes.
we mixed like kind of liposomes with the fibrils, and we did
Liposomes
the
are free lipids.
yeah, but it you know, it composed of probably both, and we did a TEM analysis, and we checked thermostability, so they are very stable.
Okay.
So we can see them in TEM, and they're pretty stable. There is another question from Marcus, right? Like about the gut-brain axis. So, what I was thinking is that we can have, you know, like we try in vitro all the different options, like to take a bunch of human amyloids and a bunch of bacterial amyloids and do a lot of cross-seeding effects between them and see which one affects the other one. So, you know, it's just an in vitro kind of an analysis, but maybe we can have a suggestion that this particular bacteria is affecting this particular neurodegenerative diseases. So, you know, to create a matrix and to just use it to raise a hypothesis for people then to check like in vivo models, like a mice model of a certain disease infected with a specific bacteria that we saw this interaction to. And then, you know, we kind of narrow it down for the in vivo people to test this gut-brain axis question. But better than that, like creating this matrix, I don't know what we can do in the in vitro level. And the formation of mixed fibrils, obviously in cryo-EM, is something that I would like to do too.
So Meytal, I also want to add to the helical intermediates. You mentioned about the alpha helix for synuclein. There's a paper by Samir Maji in Nature 2015. The first author is Ghosh. So Ghosh et al. Roland Riek is one of the co-authors. So there's a helical intermediates for synuclein as well.
So you found another one. Yeah, I'm sure that there are a bunch. I should remember, you know, the authors of these things to make a list. But I remember even at the Eisenberg lab that that we saw indicate, like I saw publications about alpha helical intermediates.
So this is scientific reports, not Nature.
Okay.
I have a question. Can I?
Sure, go ahead.
Yeah. So I was about to ask, like, in one of the slides, you have shown that the same peptide can actually form in presence of bacteria and in absence of bacteria, two different kind of polymorphism, right? I think Rams already asked that question. I was wondering, like when they form this kind of structure in presence of bacteria, so it looks like they are more oriented towards the membrane binding, right? The mechanism is more towards the membrane binding in the bacteria, right? So if that is the case, so there has been a lot of like methods that has been proposed, like in antimicrobial biology, like in the field people talk about toroidal pore mechanism, carpet mechanism. All these mechanisms are well known. So I was just wondering, like in your case, the helical fiber that you are seeing, is it because of the toroidal pore where many like this kind of structures can co-arrange, or it's a kind of like carpet model where they have a transient interaction which can induce the alpha helix and they keep on stacking? So for that, like, did you try to compare, like if you have a certain amount of concentration, let's say 5 micromolar, you started to prepare in the absence of bacteria and in the presence of bacteria, and at the end of the day you compare the thickness and the length of these fibers? So can you just comment on that?
Yeah, and I honestly don't know all this antimicrobial world with all this mechanism. You know, there is 50, 60 years of work and so many hypotheses, and I don't even want to begin, you know, challenge this field that And how do you really know, like with no high resolution information of the process, how do you really know? And I have no idea, what the mechanism, if it's a carpet-like but also I think that we need to differentiate between amorphous aggregation, just aggregation on the membrane without a specific order, and the formation of ordered fibrils. And I think it's different. And I'm not sure that they work the same. But honestly, like without the ability to actually visualize the interactions with the membranes, I really don't know. Like, so I just don't know. This is this is my answer.
I think I missed also in the presentation, like what should be the optimal concentration to make these kind of fibers? Because if you look at the biological function of LL37 in a normal human, if I remember correctly, like the concentration is somehow nanomolar per ml, like less than 10 nanomolar, right? Which
Yeah, it depends where. I think I think it can be concentrated in specific sites. But
you are right. Depending on the infection, like our body can produce more LL37.
yeah, and you concentrate it.
Yeah, but I was wondering, like in your case, like what concentration you use? Is it at range or it's very high concentration?
So for the amphibian, we already in TEM can see fibrils at 4 micromolars, which it's kind of make sense. But the sensitivity of the methods that we use is also a problem, because in TEM to actually see fibrils, you need you need really high concentration compared to other methods. And in fluorescence microscopy, you can use less, but then again, if you really want to see the foci, you need to elevate the concentration. So the sensitivity of the methods that we are using to see the fibrils is kind of the limit point here. But I would guess that they form fibrils in much lower concentration. I cannot prove it. I cannot see it. And I think in the presence of cells, it's even lower concentration. But I don't have the methods with the right sensitivity to measure that.
Right. Yeah. Thank you.
I don't know if Marcus has a question. He raised his hand.
I'm letting him in again.
There are also some questions in the Q&A folder. I'm not sure if you answered all of them. Magda, take a look at.
Yeah, I'm doing
Okay,
it.
so
Yeah.
can I ask my question? I just wanted to follow up my previous question with the gut-brain axis. I mean, do you know if we have any amyloid-like biofilms in our gut?
Yeah, there are not too many indications. Like in some, I think it's inflammatory bowel diseases, biofilms of E. coli were detected. But it's only like in specific, cases, like this inflammatory, and there are pseudomonas biofilms in the lungs. But I think are the only indications of biofilm in human, like inside the body, not on skin.
Mm-hmm. Okay, thank you.
And in implants, like when you have an infection in like hips, I think there are biofilms, but they're not very, very well characterized. The best characterized ones are the biofilms in the lungs of cystic fibrosis patients, if I remember correctly.
I was thinking about this gut-brain hypothesis for Parkinson's disease, that the disease starts in the gut actually. So
yes.
If you think about cross-seeding or so, yeah, I was wondering if there's anything in the gut which could be amyloid.
Yeah, it's true, and the flora of Parkinson patients is different than the flora of healthy people. But you don't know what's the chicken and what the egg. And you also don't know if it's the amyloids that are doing the cross-seeding, or maybe the gut-brain axis, the actual bacteria, it somehow reaches the brain, because in neurodegenerative diseases, the BBB is more porous. So maybe the bacteria has a chance to go in. So maybe it's not the amyloids, but maybe the inflammatory reaction that are causing, there are different mechanisms for the gut-brain axis, not just cross-eating.
Yeah, thanks.
Meytal, there's a couple of questions from Long Cheng I saw on the Q&A, and he's right now.
Yeah, actually, yeah, I'm like a similar question as the previous is kind of got answered, because it's any like a bacterial peptides can also present in the brain, and but I think the answer maybe is complicated, maybe also inflammation. So actually, that's my question I asked previously, like what's the potential factor in the brain, and do you think can also directly affect this structural change or lipid association in addition to the bacterial peptides, if any other mechanism? Because we know like PTM, like a post-translation modification, or the charge on the fiber core region and makes them more affinity to the RNA, which is a negative charge. So that's the protein itself or some environmental factor can directly cause this, the transition. That's my question.
Yeah, it's a good question, because we know there is a lot of work recently on PTMs, right, with tau and other, I think not just tau, I think that others, that the PTM, and there are also cryo-EM structures showing that the PTMs.
Yeah, the CBD, CBD and AD, the tau and
yeah, so they change the polymorphism, and maybe the change of polymorphism has to do with the change of toxicity. So but this is it really related to conditions? So maybe the inflammation itself can, because the sites of inflammation also have, I think it's change of pH and things like that can change the environment and maybe change the polymorph and then affect the neurodegenerative disease. I think there are different paths that microbes can change the neurodegenerative diseases, but
here there is one thing is confusing us, because it looks like this prawn-like model in the AD and the PD, because they are undergoing the cycles, intake and to secrete out and passage to the new cell. Same like the structure or some mechanism of this transition might also go to different cycles. That's if you have alpha to beta transition, maybe also have the beta to alpha transition. So also in your model, I don't know what extent the transition will be reversible or some is non-reversible.
Yeah,
for example, you added bacterial peptide, if you remove, can the transition will be back to alpha or they cannot?
Yes, so for the amphibian peptide, if we just let it fibrillate into cross-beta without the presence of lipids, it goes to cross-beta. We did solid-state CD analysis for that, but once you add the lipids, it can go back to alpha. So it's kind of a unique one, like the chameleon goes both sides, and what the strongest factor is the presence of lipids. So who knows what happens in the brain? And if you think about Roland Rick's, like, recent work with this origin of life, and if you think about, in prebiotic era, when you had, simple lipids and maybe simple amyloids, and amyloids are so promiscuous and they can self-catalyze themselves. So maybe, at the beginning of life, kind of the short amyloids and lipids, they kind of work together to form the first cells. And I'm pretty sure that there are also interactions between the RNA and the amyloids. We know that there are interactions with DNA and amyloids, like what Magda asked before. So maybe like these three components, they work together. And, there are a lot of interactions between lipids and amyloid. And with evolution, it can take the form of toxic peptides. But in early evolution, maybe they work together with the RNA to form the first cells, so there are so many hypotheses related to this, and toxicity and RNA in the brain, and I think it's a very complex system.
Yeah, and thank you so much for your answering.
Yeah, thank you. I think we're at the end of our time. John, I never asked you, do you have any questions from Meytal?
Oh, no, not right now. But it was a great talk, Meytal. Thank you so much.
Thank you.
Yeah, thank you. Thank you, Meytal. It was really nice seeing you. And I guess I'll see you again.
See me in like
In
a couple
person.
of hours.
Thank you very much, Meytal, and good luck with your lab in Germany.
Thank you
Thanks.
very much.
Bye.
Yeah, thank you so much, Meytal. Bye. Bye.
Bye.
Bye. Thank you.
You are staying back, right? We have a meeting.
Yeah, so we are using Rams the same link, or you will send me a new one?
We can use the same link. It will take time otherwise. Or do you want me to create another one? I can do that.
Yeah, maybe easier. We still, I don't know, we still have people.
Rams, can you stop the recording so that when you will end your meeting with Magda, it will say